I was obsessing over it last night. But the first one of these things is pretty much what I said yesterday. This should be brief, and then we can go on to memory updating. To really understand learning the brain, we need to know the update rules. And the experiments that said, no, they're too complicated, we're never going to get them. Are both of these statements true? And maybe that's the end of the conversation. I have my opinions, but I wonder if you guys, it's something I think that's important to think about.
Couldn't it be that, yes, we do need the update rules, but you know, there's some perspective where a lot of these different results unify and look very similar once you've got the right conceptual structure to fit them in. Like, I mean, I'm not saying this is how the brain works, but imagine the brain were some crazy deep network. If you looked at individual synapses, you might see all kinds of changes, but perhaps you could think about all of those together as just gradient descent, right? And then fit it into a unified perspective.
So the answer would be number one is not correct.
The answer would be number two is not correct.
Well,
like we will find a way of capturing a lot of information about them.
Number two is an experimental question, though. So when I say they're too complicated, we'll never get them. I mean, experimentally, we'll never figure out what they are. So the first one is a statement about theory. The second one is a statement about experiments.
Okay, let's get some other perspectives in here.
Number two is not experimental, it's a theoretical question. The biology is complicated. You have hundreds of knobs that you can turn at the synapse and get all kinds of effects. So I think the question is to what extent the theoreticians can come up with ways of conceptualizing all of these different variations in a useful way. I mean, so I think that's the main question here. It's obvious that the biology is complicated.
So I don't quite understand that, Ellie. I mean, there are certain constraints. There are certain things synapses can do and can't do. And we don't exactly know what they are. Or do you think they can do anything?
No, but I mean, there are how many people here who study synaptic rules? And each one of them and us have our own preferred way of testing these rules, and we find all of these variations around them. So experiments, the way that, at least the way we do them today, will give you a lot of diversity. Okay, and then the question is to what extent this diversity can be conceptualized, can be reduced, can be categorized, but the diversity is there. Okay, you can't.
But it's there, but for particular synapse, there presumably, this is what the picture I have in my mind, its synaptic strength, which is a complicated multidimensional thing, is a function of past input. And it's a function. We don't really know what it is. We have some hints. But and different synapses have different functions. We don't know what each one is. And we probably never will know exactly.
You will not know exactly, but there are some, as you say, okay, there are some general aspects of synaptic plasticity that we know that do repeat from experiment to experiment.
Right.
Okay, the experimenters will give you always diversity. This is what we do. Okay, we find the details. Okay, we study these to their that's your expression in the all of the gory details. And so on and so forth. Okay, so I think that the point two at the end, the answer to it will come not from the experimenters, but from the theoreticians, okay, who might find out what are the crucial aspects of the learning rules that are important for models, that are general, that are, and so on and so forth. I mean, I talked enough.
A couple more questions back there.
Completely agree with that. I think if you say too complicated, that's inherently theoretical, right?
So it's just a question back to you. I think it's for any sort of pair of synapses or pair of neurons, we can come up with an update rule. Experimentally, we can measure it. So what fraction of synapses, or at least neuron pairs in the nervous system, do you think you need to be satisfied? Like, we can probably get, in some systems we can maybe test 50%, in others you might test 1%. And what fraction do you think you need to be able to then generalize? Or do you really want a rule for every pair of neurons?
So here's what I do as a theorist. I think Bill's next. I basically start with normative rules for learning modulated by what's known about synapses. And honestly, the only thing I ever incorporate into my learning rules is locality. I assume synapses can do anything as long as it's local. I don't know if that's a good assumption. Bill, next.
So I'm not sure. I guess my worry in your formulation of item 1 is not whether it's true. It's that it suggests, right, if I tell you that in order to understand something, you need to know this thing, that does leave the impression that if I found it out, I would then understand. Okay, and yeah. You didn't say that.
I didn't say that, no.
I suspect you don't even think it, but one does have to be careful, right?
Yes, I totally agree. It's really an if, not an if and only if.
Yeah, I guess my problem with your formulation is in the first 3 words. I think a lot of our arguments are about what does it mean to understand learning before we even get to the rest of the mechanism. The example I've been thinking about a lot through a lot of these conversations is of convergent evolution. So if you were to kind of compare vision in an octopus and a mouse, right? You have 2 different solutions. I mean, putting aside the fact that vision, it may be different in, that they each have their own particular ecological context for vision, but just the problem of detecting light and how it's solved in 2 very different species, there are very different implementations at the biological level that are worth understanding in themselves to understand better each individual creature. But you could also ask, okay, what are the biological principles that underlie the detection of light that are common with different implementations in the octopus and the mouse? Like, even putting aside the modeling example, but I think the theory side of it ends up, you can view it as like convergent evolution problem, like a convergent evolution problem. So I think the issue is with understand learning. What does that mean? It's valuable to understand vision in an octopus in its own right, vision in a mouse in its own right. It's also valuable to understand learning, to understand vision in this analogy at the level of the commonalities between the octopus and the mouse.
So I agree understanding is a super loaded word, but let's say, maybe this will help, to understand how a particular animal learns. Does that help? No. Rob?
I guess I'm a bit more of an optimist. First of all, I mean, I agree with all this. I don't think we can understand the brain. But I think what you want to know about are changes in spikes. Are there more spikes or less spikes? And the details, the biological messiness, may be kind of irrelevant for the level of theory, I would say.
Yeah, I hope it's irrelevant, but
many people want to get to, right? Did the neuron, the neural circuit generate more activity or less activity and eventually make some kind of differential motor output?
Well, it's a function of its input, right? I want to know how the input output function changes.
Sure, and that can be the level simply of action potential generation. Because learning is probably expressed behaviorally. If the animal doesn't do anything different, if the agent doesn't do anything different, then are the lower level changes really that meaningful?
I don't understand your point.
My point is that, so, for example, those of us working on synaptic plasticity learning rules and caring about temperature deviations or synapse specificity and the details of, say, what glutamate receptors are activated, at some level, either the neuron made an action potential or it didn't. All those changes kind of change the spike generation properties or not, right?
Yeah, but it changes the input-output function of the neuron. And that's really what we're after, right? When learning is really changing the input-output function of a neuron, and it'd be nice to know, it's sort of helpful to know how that happens.
And if your level of description is in terms of the spike rates, There's still input-output functions for
Yeah, the
neurons and
output's
neural
spike
circuits.
rates,
Yep.
and the input is PSPs. And how does that mapping change over time with learning? And it'd be super nice for theorists to know what that is. We don't actually have to have it, but eventually to make contact with biology, our theorists have to be consistent with what's observed. And what's observed is super, super complicated. Look, like I say, I don't think this is an insurmountable problem, but it seems like what came out of the conversation yesterday.
Let me make one quick last point, which is the question of where can be really complicated because a lot of things can change over different timescales. But I think when is maybe more fundamental because there probably are some things that change first, the earliest detectors. And finding those, I think, helps kind of constrain this problem then. If you can find kind of the earliest form of learning when the state has changed, then you don't have to necessarily study every synapse and every neural system.
Yeah, I'm trying to get back to this question of like, what do the experimentalists need to do to get the basics, the most important info. And so in our system, we were seeing things at the spiking in vivo level and behavioral level that we couldn't make sense of given what had been described in the rules. So we went and re-looked at the rules, and we found a different thing than people had assumed was there. And I'm wondering, but we have so many advantages in our circuit for doing that. So I'm wondering how to do it in more complex contexts. And it seems like a lot of times the modelers say, okay, we're going to assume simple rules because we want to we want to use the STDP rules everybody's seen, but there's not a lot of modeling work that says, okay, let's hold all the other constraints constant and see what happens as we vary the rules, and that might help identify some interesting directions in parameter space for the experimentalists to go after. And I'm not sure if that's what you were saying, there could be something that comes out of modeling that could provide some structure on our thinking about this.
So what you're saying is
work is happening in that, to get us there. Is it happening?
So theorists can reduce the parameter space.
You said it could happen, but is it happening?
Yeah. So maybe I'm not sure this is exactly what you're saying, but there is work trying to meta-learn good plasticity rules for spiking neural networks in the deep learning tradition, right? So what comes out of that could be immensely complicated, but maybe it gives you some higher level way of thinking about all those mechanisms because they're serving the function of the overall system, for instance. Yeah, and then, so I think maybe there's a direction there. And then just the other point I was trying to make was just, things look complicated until you see it through the right theoretical lens, and then maybe there's a way of putting all these observations together, which we just haven't gotten to yet. Yeah.
Sorry, this is not going to be super constructive, but I guess I fail to see how you couldn't have written exactly the same 2 points about just about anything about the brain. To understand blah in the brain, we need the blah information about the brain. 1, 2, biology is too complicated, so we'll never get it. So what is there anything specific, and this is the serious part of the question, do you think there is anything specific about learning and plasticity, synaptic plasticity here?
So no, I mean, your point is absolutely correct. For me, learning is I think one of the more tractable and interesting questions in the brain, but you're right, you could say this about anything. And like I say, I'm not sure I believe 1 or 2. I mean, I think 2 is kind of true, but I'm not sure I believe 1. Bill?
I'm not sure I actually agree with that.
Oh, you think?
There's something special about learning, but it's a derivative. Right? There are moments in time where something has changed. So I think those moments in time really can begin to figure out something different, as opposed to what's the neural code for this? Which it's difficult to know when it even begins or ends.
Bill?
Yeah, I guess I'm going to echo Mate's point that I'm in the background of this. There's this issue of, what level of detail we need. And, I do come from a tradition that says you should be able to smooth out lots of details and get simple things. But there's a problem with that, which is that experimentalists don't have, so in physics when we do that, or in traditional areas of physics, there are experimental knobs that correspond to our phenomenological parameters. And actually you need a chemist to manipulate the microscopic parameters. So if I want to study electron conduction in solids, right, I don't have to think about atomic structure. I can apply a magnetic field. And my phenomenological theory knows what to do with the magnetic field. In biological systems, there's been this explosion of ability to manipulate things at a very microscopic level. And on the other hand, if I give you a phenomenological model and I ask my experimentalist friend, could you change this parameter and tell me what happens? He says, I don't know how to change that parameter. I can go in and, my colleagues and I also spend some time thinking about genetic networks, right? I don't know how to change this phenomenological parameter, but I can go into the regulatory site and switch an A to a G, right? And I can do that a million times, right? And I can do all sorts of fantastic things based on that. So I think there's an issue when we talk about levels of detail and, what do you mean by knowing the rules? That you want to be sure that theory connects to the kinds of experiments people can actually do. And when that landscape changes very dramatically, we should pause. I mean, I think we should be worried about the fact that the first great discovery is that neurons generate action potentials, and most of our model neural networks don't have spikes. The next great discovery is that the dynamics of electrical activity is based on ion channels. And this, by the way, is, the most quantitatively described example of functional protein dynamics in all of biology. They're gone from our models, right? This is not a good trend. I'm not sure I know what to do about it, but I think it's something we should worry about.
I'd like to agree with what's just been said, but maybe add to it. I think you first of all have to ask who wants to learn, who wants to understand learning and why, and for what purpose. If it is to help design models and to create artificial intelligence to replace ourselves, that's one thing. Is it for medical reasons so that if something goes wrong and we know how to fix it, that's another imperative, and you might want to know different sorts of things. I think I'm one of the great guilty ones here in this meeting who said number 2, that the biology is so complicated that, we'll never get all the rules. But depending on your purposes, and I think this is kind of your point, you might not need to, you might be able to smooth over a lot of things. I tell my students, every protein sitting on the membrane is a plastic protein, whether it's in the synapse or not. And you can change as a result of activity with God knows what, based on what rules, but you can induce a change in the expression, the phosphorylation state, whether it's on the membrane or inside the cell. Everything is plastic in the cell, basically. So yes, the parameter space is huge. But you can smooth across all that as needs be for the purpose for which you want to understand learning in the brain. And if we go to like LTP, the great exemplar, we have a very good idea what the fundamental rule is for a certain synapse, Schaefer collaterals to CA1, for example. But then there's layers upon layers of modulatory elements that will influence. If you just think about the activity levels pre and post synaptically, there's a rule about that, but then the actual outcome is mediated by layers upon layers of other complexity in the cell. So, and that might be important if you have a memory disorder of a specific part of that layer that you might be able to fix, but maybe for models it's not so important. So I think it comes back to for what purpose you want to understand learning.
So let me briefly answer that. So I have a confession to make. As a theorist, I smooth over lots of stuff. I have to, but because of that, I don't pay that close attention to the experimental literature, because it's full of these esoteric facts. I kind of, okay, we can have metaplasticity, short and long term, so I know that kind of thing, multiple rules. But the details, in some sense, there are so many of them, it's daunting, and I ignore them, which is maybe a bad idea. I don't know.
So I just want to, I guess, repeat myself in some sense, but kind of make an appeal again for at least the possibility that a sort of mesoscopic scale of explanation will be possible. So, I mean, we've seen, we have a lot of very sophisticated, very predictive, quantitatively predictive behavioral models, right? They are describing the brain, but not at a level that makes any reference to neurons whatsoever. And so this, the only reason we can do that is because we're looking at a system that has evolved to actually function at that level. It's plausible. There's no proof here, and it may turn out to be impossible, but it's plausible that there will be an explanation that's self-consistent and quantitatively predictable in the same way at the level of neuronal activity, right? So I might be able to say, given that the network has had this level of activity in the past, there's some learning rule at network activity level that tells me how its dynamics is going to evolve in the future. Of course, that's built on changes in synapses, but it may be self-consistently described, and perhaps somewhat more simply described as behavior in many cases seems to be, at that intermediate level, that sort of mesoscopic level. Now, if you define understanding as having linked it all the way down to the biochemistry of the synapse, then yes, we will need all of those details. But I'm willing to sort of, look at these different levels of understanding as we work down. Ultimately, ideally, they do all link up together, right? So the neuronal dynamics underlies the behavior, and we understand what's going on with that, how to link those two. And the biochemistry underlies the population activity, or the functional activity, and we know how to link those two. But we can spend a lot of time trying to understand each one of those at its own level, and that is a kind of understanding. Right? An understanding of learning. Just like behavioral, understanding predictively, learning and behavior is an understanding of learning.
Okay, so what you're saying is we can skip plasticity and look at evolution of activity and write down rules for that? Okay, yeah, no, I agree, totally agree.
Ultimately, of course, we do want to know
right. I don't think that'll be possible, but if we could, I think it would be. It's certainly an understanding, a relatively deep one.
Okay, this has been a great discussion. Can we invite the speakers from the session up too?
Yeah.
And
sorry for derailing that. Like I say, I was obsessing this 2 in the morning.
Yeah, we can carry on discussing this, but we can also open it to questions for the speakers as well.
I just want to share kind of one observation that I got from watching the talk shows the last few days is that it seems you cannot look at activity alone in order to be able to solve this problem. It seems you need to have some understanding of connectivity and some understanding of other aspects of anatomy, such as glial cells and things of that sort. And that if these are not at all present in the model, I don't think you'll solve it just from looking at spike rates.
Yes.
It's still about this, or should we move on? Is it okay to continue? Yeah.
Yeah.
Okay. Yeah, just to add one maybe slightly optimistic point about this. I do think actually I think you want to have realistic models of the brain. You do need both the update rules, but there's no point of knowing the update rules unless you actually know the connectivity, so you know which connections are. So you need two things, the connectivity and the update rules. But I think maybe things aren't as hopeless as they seem, which is one thing we've learned from connectomics. When you sort of look at just portions of a circuit and not a complete circuit, then things seem like a complete mess and incomprehensible. But once you look comprehensively at an entire circuit, you get a lot of simplifying principles. There isn't an infinite number of circuit motifs. There are relatively few. And I think probably that if we looked at somewhat comprehensively at these update rules across a large set of neuron pairs, then we would get a lot of simplifying principles, and there aren't infinite numbers of them, but that's just my feeling.
Hi, so I actually wanted to ask Mattia a question about kind of thinking about neural implementations, right? So your kind of belief state model, you know, because you're weighting the update based on the posterior probability of each context, and the contexts are discrete, you have this discrete probability distribution, right? Which seems kind of different from the other cases where people have thought about neural implementations of like sampling or probabilistic population codes. And so do you or have others thought about neural representations of discrete probability distributions?
So a couple of answers to that. So you can, so in principle, I don't think they have been worked out in sufficient detail to really kind of make contact with neural data, but you can certainly extend a lot of existing frameworks, including sampling, and you know PPC, at least PPC-like codes that encode discrete probability distributions. And in fact, in some sense, one could even argue that, for example, as far as sampling goes, the best evidence is about kind of discrete switching. In some cases, you know, in the hippocampus, switching, you know, the flickering phenomenon looks a lot like it's suggestive of a kind of a sampling-based representation of context. So that's one thing we discussed in the annual reviews paper that we wrote about this. Also, not in the context of context, but you know, in cases of perceptual biostability, the way neural responses also seem to kind of switch between representing two percepts is also seems to be consistent with the sampling-based representation, and people have actually made arguments about that link there, including Sam Gershman and others. And the other frameworks are also extendable. I don't think the details have been worked out in an awful lot of detail in any of those cases. So that's part number one. Part number two is that contexts themselves might not need to be entirely discrete either. And in some cases, you might be interested in inferring a continuum of contexts that might be hierarchical. You know, contexts might be hierarchically organized. So we really kind of, in some sense, deliberately took the simplest possible representation as a starting point, which is like a flat, non-hierarchical, discrete case. But that doesn't, you know, if pushed, I would not bet my life on that. Let's just put it that way. So I think there might be convergence in both ways. But I think, just to make sure, I think contextual inference, I think more generally, I think your question implies that, and I just want to reinforce that I agree with what I think your question implies, which is that contextual inference might be a very interesting use case of working out how probabilistic inferences are actually implemented in the brain. Is that what you meant? We agree.
A question to all three of you. Retrieval is an integral part of memory, and I think especially in the updating of memories, it also plays a big role. Do you think there is a direct relationship between memory reactivation and memory retrieval, and whether it's updated or not?
I can start with that. Yeah, I definitely think, I mean, at least according to all the historical literature, there's some sort of retrieval event that has to occur for that updating to occur. But I think something that, as we talked about earlier, like there are all these boundary conditions, like retrieval alone is not sufficient to drive memory updating. There has to be something, either the animal's not trained very well and hasn't fully learned it, or a change in the timing, or a change in the context, or something to actually drive that updating and that reconsolidation process. Like just retrieval alone is not sufficient.
I'm going to take that in a slightly different direction, which I've at least struggled with, and especially thinking about how we organize things together, right? Retrieval is, in at least animal systems, is measured by expression of a memory, but I can ask you to think about your favorite day and you are thinking about it and retrieving it, and I have no measure of it. And we do struggle with that when we're looking at retrieval of certain kinds of memories and how they're either getting upgraded, reactivated, or any kind of modulation. And I don't know if I at least have a good experimental handle to be able to test that and then make predictions about that, especially in animal models.
So just to make sure I understand the question, the question is whether memory retrieval is necessary for updating. Is that the question?
Whether it's parallel, whether the retrieval implies that.
Right. So, so the model that I talked about, I don't think it makes. I think that's a very interesting question, and it's fundamentally, in my mind, that's an algorithmic level question, and our model is not really, although it's easiest to explain as if it was an algorithmic level model. It really isn't. It's much more of a computational level model in terms of Myers levels of analysis. So what the model does say is that fundamentally the same inferences about the same ongoing inferences about what context is relevant when should be controlling both of the memory updating and retrieval as well as memory creation. What it does not say, does not specify, is that at the level of algorithmic detail, whether you actually need to, for example, causally retrieve a memory in order to be able then to update it. I think that's a very interesting question. It's just not something that the model as it is would currently make any prediction about.
In your model, you had the subject doesn't need to be in a context for the contingencies of that context to keep on updating themselves. If there's some expectation that they will have been updated, as the subject goes back into that context, reactivating those contingencies implies a retrieval, right? So knowing that these contingencies have changed over time implies a retrieval.
Yeah, so in general, it is true that according to the model, you should be updating your estimate of the contingencies of all the contexts at all the times, except when you're absolutely sure that you're not in that context. But in general, your probabilities over the context will never be exactly zero. So there should be at least a little bit of updating going on at all times for all your memories. So that's true.
Thanks. This is also for all three of you, in terms of how you're thinking about how context is constructed. I'm thinking about how animals or humans sort of sample their environments and how quickly they can kind of recognize where they are. What do you think they're attending to, and how quickly can a context kind of be assessed?
I'll answer, even though I'm the only person who didn't really talk about context. I mean, there's a lot of experimental literature.
In your case, I meant object recognition doesn't require 5 seconds.
Right.
How quick is human visual object recognition?
Yeah, absolutely. And I mean, there's a good amount of experimental work, at least on context, and you have to have a certain amount of time to actually build that configural representation of a real context memory, right? So you put an animal in a fear conditioning box and you shock him right away. They don't, in theory, I mean, they don't learn that context is predictive of the shock. You have to have enough time to build this configural representation that then gets paired with the shock. Otherwise, you could maybe learn where the light is, that light is associated with the shock, but the actual configural representation does take time. How long that time is probably depends on a ton of factors, the animal, how complicated the context is, etc. But I think in all cases, to have a real configural, bona fide context representation, it definitely takes more time than something that's super discrete, like a white noise cue.
So I think that's a really interesting question, because a lot of times we talk about, at least in hippocampal or retrosplenial systems, spatial and contextual memories very synonymously, and they're clearly not, right? I don't need to sample this entire room if I have to come in tomorrow through the back door to know I'm in this room. So I've probably never made place styles to a lot of these places, and there are some interesting studies that say that this recognition of what the context is happens really early on. It doesn't require, it requires time, but it doesn't necessarily require you to sample your entire environment. And yeah, I guess it has some interesting implications for how synonymously we use these terms, but also for the experiments we design and how we interpret them.
So one insight that that our model provides about this question, I think, which at least to us is quite important, is that I think that question is really two questions. One is that, so two things need to happen, right? You need to be able to infer that you are in a different context than where you were previously in the previous time step or in the previous trial. And then also you might, for whatever context you think you are in now, you need to be able to update the memory associated with that context. And those might happen on very different timescales. So, for example, in the model learning experiments that I told you about, but I didn't tell you about the particular set of experiments that we also did. Daniel will tell you about more about those later. So there is an experiment, for example, where we don't even have distinguishing sensory cues that will tell you which context you are in. It's just the motor feedback that you can use. But once you have learned that there are two different contexts, there is a particular experiment where essentially we can switch your inferences about what context you are in two trials. In fact, to largely already in one trial by providing the right feedback. No sensory cues, so nothing that will overtly tell our subjects that they are in a new context. And it turns out that there are close analogs of that in the animal conditioning literature as well. I think they're called reminder trials, Peter.
I think actually that's kind of what kind of inspired my question. Contextual inference is sort of like the scientific method. So I'm wondering if in the context of a single trial, are the human subjects in the beginning kind of sampling the space to try to get a sense to perturb it, do experiments, to figure out what context they're in? How are they sampling that?
Just a big comment on the updating versus retrieval. So in your model, and I say, because context could actually have systematic change over time that you would know about, even if you're not even observing them. So in fact, you do expect there to be updating without any retrieval at all, because it should just carry on by itself, right?
Yes, but it's so you are, yes. So you're right. Yes. So the feedback, there is spontaneous evolution of things independent of feedback. Yes. And we see signatures of that actually in some of the experiments.
Well, we're in a loop, so this is also a question for everyone. So we tend to think of context as multisensory, like the integration of multisenses, right? But sometimes I'm thinking about a specific kind of a ratatouille event where the food critic tried this food and it immediately brought him back to his hometown and his mother and everything. So I'm thinking about, so how do you think memory updating and dendritic plasticity work in the sense of one single cue being so salient that it can evoke a context? And in the case of modeling work, are these models allowing for these kind of events to happen?
I have to think about this. And I think that was kind of my response to the retrieval memory part, was sort of thinking of the kind of questions. Let me think about if I can say it correctly. In some ways, what you're talking about is a very intuitive form of linking of events, right? Like we have a smell that's really associated with a context, and in our particular case, I'm showing you two contexts and they're the same modality, they can be linked. Obviously, we link different modalities all the time, like you're talking, and olfaction is a very powerful way of doing it. So how do we link cross-modality is, we don't really know. My guess would be it's very similar mechanisms, perhaps in parts of the brain, like I was referring to Jennifer's question, that where these inputs can impinge on the same neurons naturally would be a better place to do these cross-modality linking events. And a lot of, I think, our linking, at least in humans, right, is very retrieval based. We don't necessarily have to be put in a context to rethink of something triggers a recall of some other event, and that's actually what causes you to link. And there is some experimental data to show that you can encode whatever stimulus weeks before, have a really small retrieval event. It triggers plasticity. That's been another study, and that retrieval-based event can also trigger linking to a subsequent future event. What are the boundary conditions? I think would be probably something that we'll figure out over the next few decades.
I don't have much to add except to say that, we know emotional memories are super salient and often very persistent too. So that probably plays into, if you have like a childhood memory or something you remember really strongly, or I'm guessing more likely to link that as well, like an emotional memory, like a fear memory versus, something like object location memory. Those animals forget that usually within a week, even with additional training sessions. So I think the emotional content and the stress that you're going through at the time makes a huge difference.
Yeah, so we actually had a very interesting discussion about this just now in the coffee break about kind of comparing notes across very different kinds of paradigms, including motor learning and more kind of explicit forms of learning. Flores told me about his experiments, and so first there is the observation that yes, some kind of sensory cues seem to be much more potent in terms of allowing people to distinguish between different contexts than some others. But then it doesn't stop there. It's also the case that it depends on the domain of learning. So for motor learning, some cues are very potent, some cues are not, but the same cues that are not potent for establishing different contexts in motor learning are actually quite powerful in Flores's hands in the kind of learning paradigm that he uses. So I think there's a lot of richness there. If the question is whether our model can capture that, in some way, at a minimal level it can, because essentially it learns the distribution of cues that is associated with each context, and being a proper Bayesian learner, it has a prior over those distributions as well. So if the prior is such that, a particular kind of cue is completely uninformative, then that cue will never be able to be used to distinguish between memories. I think that's way too simple. I think it's more, there's more richness to how things actually work than what the model would be able to capture in its current form. So that's for future work. But I think but I think the general idea is that we probably have a lot of evolutionary built-in priors that tell us what sort of cues in general are going to be relevant for distinguishing between what sort of contexts, like what kind of cues are important for distinguishing between motor contexts, what kind of cues are relevant for distinguishing between spatial contexts, and then we can learn within the bounds of those evolutionary kind of specified priors. That's, I think, the way we think about it.
Hi, Mate. I have a question about your study. So I was wondering if all the memories are being updated simultaneously. Is that putting like a lot of computational intensity? That seems like that would be very computationally intense and also introduce a lot of simultaneous instability into the system. So I'm curious, how do you explain that?
I'm not sure what you mean by instability, but sure.
I guess if everything's changing at once, then there's risk for I don't know.
Yeah, that's how life is. I really don't think. And the brain has to cope with dynamic instabilities all the time at all different levels of organization. As it happens, we've been quite interested in some of those dynamic instabilities at a different level of organization, at the level of network dynamics. But I mean, let me turn that question around. Do we really think that there is such a thing as a frozen, crystallized memory that is just there for you as a book on the shelf, and unless you touch it, it's unchanged? Do we really think that? I don't know. I would find it hard to believe that that's really the case. Well, that's not my problem then. No. So I think there are different timescales for changes in memories, and sure enough, there's going to be domains where you think, and so this goes back to Peter's question. So if you have inferred that in a particular context, kind of the dynamics, the ongoing dynamics of the contingencies are really, really slow, then unless you get specific new information that something has changed, you will assume that things are the same as you left them last time. So it's not impossible, but I think there needs to be good reasons to either, again, evolutionary built-in reasons for a particular kind of context or a particular kind of information, or there needs to be ample previous actual experience to tell you that yes, things are really, 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 really stable in this context. But yeah, I mean, I'm kind of embracing the view that all these memories are being updated at all times. I would just love to know how that actually happens and how we do this, uncertainty-weighted multiplexing of updating. That to me is a big open question.
I have a slight response to the Ngram cell. No, it's actually, I think there were obviously like these static ideas, right? But it's pretty clear, and we've talked about it a little bit, that representations even within Ngram cells drift pretty rapidly. So I think at least hopefully a modern evolved understanding of it is that those were, across days, these engrams are evolving, and hopefully for a purpose that's very similar to incorporate newer information of the world. But I think it's somewhat consistent.
I also think the very act of retrieving that engram or that memory is, in name going to change what you're storing. So it's like kind of impossible to tell if that memory is static, because just the act of retrieving it changes it.
Let me just actually go back to Peter's comment. So it's true that in the model, there's no difference in terms of updating beliefs about contingencies, whether in the presence of an observation or in the lack of an observation. But I think, again, once to consider like more algorithmic level issues, I think a lot of those spontaneous changes that you were referring to will look a lot like just decay or forgetting, whereas the updating that I had in mind when I answered the previous question was more like actual updating with new information. And I think it's an interesting question of, how much at the level of mechanisms, how much is shared between those two kinds of updates, if you like.
Okay, I think we're coming up on time, but I just want to close with one last question to ask if you guys can explicitly bridge your presentation. So I was trying to think, what does the coin model mean? Like in your context, you show there's this temporal window where the linking happens. Would the basic coin model show that? That wasn't clear to me. So yeah, do you have any thoughts?
I guess that's fine. So for sure, so the coin model definitely has sensitivity to kind of the temporal evolution of contexts, and it can show that, but it's in some sense if that's the single data point that it needs to fit, then it's a way too complicated model for that. It can overfit that, but that to me would not be really satisfactory. So the basic mechanisms are there, mechanisms in the model are there to capture this kind of phenomenon. But I think it would be really interesting to, and that's actually something that I'm personally very interested in, is taking this model to model actually these kind of experiments and see whether and do the kind of thing that is routinely done in a lot of the human behavioral study literature, that is fitting these fine-grained behavioral models trial to trial, but actually do that, and of course some of that has been done for animal studies as well, not so much for contextual learning, I think, but to do that here, and I think that might have a lot of potential for then starting to reveal also the neural mechanisms, because once you fit these models trial by trial, you can then look under the hood, see what's going on trial by trial with the internal representations of these models, and try to kind of relate them to whatever you see in neural activity and how neural activities or neuromodulators change from trial to trial.
Just want to say from like a neural mechanisms, like a molecular mechanisms perspective, I'd love to see mechanisms that are conserved across different tasks that are doing the same thing. And something that I didn't mention before, but we talked a lot about transcription and translation, new protein creation, new mRNA creation being really critical, but something that's also really important for whether a retrieval event initiates reconsolidation is whether degradation is initiated. You have to actually destabilize things before you can modify them. And I think that's a real important key when it comes to memory updating, kind of across levels of analysis that's kind of underappreciated as well.
I guess so. I just have that easy task of saying that linking is like really, really similar to updating in some ways, right? So I kind of just focused on one behavioral aspect where you have a context that you saw before and you freeze on that too, even though that's never explicitly fearful. Another thing that we showed in the original paper, and you kind of notice if you really paid attention to even my data today, is it doesn't just cause increase in freezing in the context that they've never been shocked in. It also drives up the freezing in the original context that they do get shocked in. So it doesn't just allow you to link a memory, it also updates I guess that's a loose way of using that word, but it also is changing the original memory and the strength of that memory. It makes it stronger. And I guess in my worldview, you go into a new, a box that you're going to shocked in. You're going to be like, hey, this is the box I saw previously. And right before that, I saw another box. You're retrieving both those events in order to be able to link them together and updating these events as well. So how that all ties in together would be, I guess, interesting.
Great, thank you. All right, I think we're out of time. Thanks very much to our speakers. Let's give them a round of applause. And also, let's thank again Jennifer for organizing this amazing conference. I don't know if you have any.
Yeah, I just wanted to make 2 announcements. As you know, the conference is part of a wider, longer program on statistical learning, and we always have a barbecue on Thursday. So I wanted to extend an invitation to whoever of the conference participants is staying here until tomorrow. This will happen at 7 p.m. in the Munger residence, which is where the program participants are staying. So let's say I'll come out to the front door at 7:15, and whoever is there, I'll bring them in. We offer wine and cheese, and usually we bring our own food to barbecue, but I'm sure all the program participants will be happy to bring a little bit more and share their food with whoever is there. So that's one thing. The other thing is, which is maybe what you want to say to well, as part of the program, we thought it would be very relevant to have information on what are the tools that the brain is using to do learning. And this conference has been absolutely amazing, and I wanted to thank Jennifer for an incredible experience, a fantastic choice of speakers, and also the format of the conference, I think, has been fantastic to have so much time for discussion, to have this idea of having the slides that people could bring in for the discussion to bring more data and more wood into the fire. I think it's been really incredible, so I wanted to thank you. Really, it's been incredible.
Yeah, so I also have a number of thank yous. I mean, first of all, I think the program really set the tone for this conference, and kind of the level of interaction is possible in part because it's part of this other thing where people were spending enough time together, as I was describing to some people not from the group here, to get over the politeness and just to start to say like, well, what about this? I really disagree. And, I think that's awesome. And I think we need more of that in science in general. And so I really appreciated the culture that was set for the conference by the program. I really want to thank my co-organizers, Livia, Mate, and Joseph. I sort of, they were doing so much for the program. I did a little bit more for the conference, but we really worked together to think about how to make this happen and how to have it be something different that I hope all of you found useful. I really appreciate, and the KITP staff, if you see them, please thank them, because they are really pros at running conferences and doing it smoothly. So the whole staff, but especially Claudia Gutierrez, really deserves credit for making this work so smoothly. I want to thank the session chairs. I want to thank all of you who fought all of the travel challenges to get here, and for stepping up and offering perspectives and really being interactive and offering up challenges to everybody. So thank you all. There is lunch served. There was some question about that. We do have lunch in the courtyard today. And then I hope to see a lot of you at the barbecue. I'm wondering if we should be a little more organized about the food. I would be happy if people want to email me if you're sure you're going to come. And then I can go to Costco and get some extra chicken or something to put on to make sure, because I want people to come and not feel pressured to bring anything. So if you're coming and you don't think you're going to be able to make it to the store, or you don't feel like bringing it, just let me know, and I'm happy to help out with that. And let me write my email on the board so you can do that. And I will have a car, and I will go to Costco sometime later this afternoon. So earlier that you tell me that you're coming, I can plan. Oh, and the Munger, yeah, the Munger residence. Yeah, I can just send it to everybody. Okay, yeah. All right, thank you all. Enjoy lunch.